Regulatory Science Symposium, organized as part of the 18th session or edition of the Dubai International Food Safety Conference. We're pleased to welcome you all, both here in the room, so many of you, and also online for colleagues who are joining us via the webinar system. We're also fortunate to be broadcasting live on YouTube on the channel of the Global Food Regulatory Science Society. I just wanted to acknowledge the support of the organizers of the Dubai International Food Safety Conference for enabling the organization of this symposium, but also the scientific partners of these sessions, the Global Food Regulatory Science Society, the International Union of Food Science and Technology, as well as the Platform of Food Risk Analysis and Regulatory Excellence of Laval University at Université Laval in Quebec, Canada. I'm sorry, I'm just adjusting because this is speaking at the same time, so maybe if you can... Let's see if it stops speaking. Apparently it's not. We're good. I think we're good, yes. Sorry, just adjusting some technical difficulties. So this session is actually dedicated to food risk assessment, and we're trying really to give some illustrations of the importance of risk assessment in framing the decision-making process, obviously taking into account the realities of the Arab region. So we're going to be taking some examples of the Arab region. We have a number of speakers and a number of examples that we're going to be going through, but I will start briefly with a short introduction and putting in context essentially the talks and the presentations that will be made. I'm going to ask actually colleagues to put the presentation. So, risk assessment is really an important activity of food regulatory bodies, and we know that regulators, food regulators, are the ones who are making the decisions and have, in fact, a delegated authority that is given to them generally through the food legislations and the subsequent regulations to make these particular decisions. So they are empowered to do that. Now, of course, when regulators are doing that, what they are trying to do is really to manage the risk, meaning to minimize the potential risks that are associated with the consumption of food to consumers. And the decisions that are expressed by regulators are not necessarily only in a regulatory form, meaning a decision that is enforceable. Some of the decisions or some of the interventions can be voluntary interventions. They can be in the form of guidance. They can be in the form of advice, whether be it to consumers or to the regulated industry as well. Of course, in order to ensure that these decisions are robust, the decisions are trustworthy, we need to make sure that these decisions are based in accordance to a logical framework for decision making. And that's what food risk analysis is. Food risk analysis is a decision making framework, is a methodology that is followed by food regulators that ensures that these decisions, because they are following a methodology, are less likely to lead to mistakes, to errors, to issues. But also, because they are following this framework, they are leading to building the trust that we need, meaning they are leading to making the decisions better understood. They are leading also for these decisions to be potentially trusted as a result of sharing all the information upon which the decision is made. And in fact, this logical framework was advocated by FAO, WHO, and particularly by the Codex Alimentarius Commission, and has been adopted by Codex in 1995 as the basis for all food safety and nutrition decision-making processes. This logical framework that we call food risk analysis is composed of three elements: risk assessment, which is the scientific component, risk management, which is the policy based component, and risk communication. And risk communication is, in fact, not the mere communication of the risk, but rather the process of interaction throughout the entire decision making steps, the process of engaging with all the partners in the decision making process. So it can be between risk assessors, risk managers, but also all the stakeholders. So that includes industry, consumers, but also academic partners as well. Now, a functioning food control system is generally a food control system that, of course, relies on a robust legislation and regulation that gives the empowerment, but where the decision-making process is really built on these three pillars of risk assessment, risk management, risk communication, and of course, where things are holding together thanks to a good governance and collaboration between partners. Now, what we see generally of this process, the visible aspects of the food control systems, is essentially the decisions that are made. All the other steps are not necessarily as visible. What we see from a regulation is the decision that the regulator makes, or the communication that the regulator makes. So that's actually the most visible part. It doesn't mean that the other parts are less important. In fact, the other parts are equally important to justify the visible part of this process. And that's what risk assessment is for. And in fact, risk assessment, or following this approach, is not necessarily a discretionary position from regulators. Countries that belong to the World Trade Organization, meaning countries that are signatories to all the treaties of the WTO, particularly the SPS agreement and the TBT agreement, SPS, that is the sanitary and phytosanitary agreement, are bound by these clauses. Article 5 of the SPS agreement clearly indicates that regulators cannot be making a decision unless they base it on the assessment of the risk. And of course, they define that as an assessment of the risk to human health. So I don't know if this pointer gives me the right direction there. It's not going. Oh, it is. So the risk to human health, but also to animal and plant health for the other parts. Of course, for food safety, we are focusing on the risk to human health. And this article also explains that we need to base this on the available scientific evidence and on the appropriate methodologies to do that risk assessment. It also directs regulators on the way they will be selecting the risk management approaches and then applying these risk management approaches, mainly with the attempt to minimize impediments to trade and by ensuring that their decisions are not arbitrary and are not discriminatory. They're not discriminatory, meaning that they do not make a distinction between something that is made domestically versus something that is being imported from another country. Now, the manifestations of food risk assessment, and this is really now the entry into our discussions, the manifestation of the risk assessment is really the support to any type of risk management decision. These are examples of risk management decisions. It can be the development of a standard. When I use the word standard, I use this with the definition of codex. What does this mean? A standard can be, an example, a maximum level for a contaminant in food, a maximum residue level for an accepted pesticide, a maximum residue level for an accepted veterinary drug, for example. But it can also be the development of a guidance or a code of practice that can help producers improve their conditions of control for a particular hazard or a series of hazards. It can be also a series of advice to consumers. The example that I gave is fish consumption to pregnant women. That's a risk management approach that is not a regulatory decision. Nonetheless, it is a risk management approach that comes from a risk assessment. And those type of decisions generally are what we call preventive risk management decisions, because when you set these requirements, you're trying to prevent an incident from happening. But you have also the risk management decisions that are reactive decisions. They are reactive to an incident happening. And the example is managing a recall or mandating a recall. Generally, arriving at a decision to recall a food product is stemming from a risk assessment associated with that particular incident. And again, we're not going to get into the theory of the risk assessment. You know that risk analysis in general is an iterative approach. Risk assessment is the scientific part of that. Of course, risk assessment, being the scientific part of that, requires investments in competencies and in capacities, and that's why we need to leverage international knowledge, and international knowledge provided mainly by the expert bodies to the Codex Alimentarius Commission, the Joint Expert Committee on Food Additives, the Joint Expert Meeting on Pesticide Residues, and so on. So JMPR, GEMRA, etc. The question is, can I use all the information on this and can I basically copy paste a risk assessment? And the answer is, to a certain extent, it's not copying and pasting. It's rather understanding and adapting the risk assessment. There are elements that generally can be adopted, and those elements are pertaining to the hazard characterization, because we consider that the characterization of the hazard or the impact on health is really an impact on human health. So there shouldn't be a difference between somebody living in Egypt and somebody living in Canada. We are all humans. And the risk assessment takes into account the disparity between, let's say, the vulnerable populations, if we need to add some level of protection. So the risk assessment consider that, meaning the hazard characterization consider that. Where the differences still persist and where there will need to be some assessment at the local, national, or regional level is to estimate the exposure. And the exposure requires two key parameters. First of all, the occurrence of the hazard that can be really associated with the region and can be different from one region to another, can be also based on the practices themselves, and also the consumption. Food consumption can be different from one region to the other. So geography matters. And as a result of the fact that geography matters, it means that we all collectively need to continue to invest in risk assessment capacity, and particularly in exposure assessment capacity. And this, of course, requires the investment in competencies, the investment in availability of data, and the investment in some of the enablers, such as the tools, the softwares, anything that will allow you to support those two other parameters, meaning competencies and data. That's what we're going to be trying to see some illustrations for during this session. And we're going to be starting, in fact, with the first illustration from the Arab region. This is coming from a very recent publication that was made available in the scientific literature, in fact, this month, where essentially a collaboration between teams, scientific teams in Lebanon and scientific teams in Canada conducted a risk assessment related to the occurrence of aflatoxin M, and particularly aflatoxin M1 in milk. And this will be an illustration where some of the outputs of the risk assessment can be actually reassuring. So I will be leaving now the way to my colleagues, Dr. Jérémy Théolier and Dr. Sylvia Dominguez from Canada. Of course, it's actually, I think, 2 a.m. in Canada right now, so we did not ask them to be with us live, and they kindly actually recorded their presentation last night and shared it with us. So we will watch this presentation together. Thank you.
Good morning, everyone. Thank you for joining us today. My name is Jérémy Théolier. With me is my colleague, Dr. Sylvia Dominguez. We are happy to share our knowledge of risk assessment and how it can be used to identify vulnerable subgroups in your population. So, first of all, I'd like to point out the fact that this presentation is based on a collaborative effort involving 3 teams with different areas of expertise, namely Dr. André El-Houri's team, which produced the occurrence data, Dr. Maya Otet's team, which contributed with the consumption data, and our team, so Samuel Godfrey's team, which was in charge of the risk assessment part of this work. So we published a scientific paper in October 2024, so it was really recent, in Food and Chemical Toxicology Journal. It's an open access, so people can have access to the paper for free if they want. So let's start by understanding what's wrong with AFM1. So aflatoxins are toxic secondary metabolites produced by some Aspergillus species which colonize crops, including many dietary staple foods and feed components. Among them, AFB1 is the most prevalent and the most toxic one. AFB1 is metabolized into AFM1 in the liver of individuals and then excreted into the milk of lactating mammals, including, of course, dairy animals. The presence of AFM1 in milk is a worldwide concern, as even small quantities of this metabolite can be significant in the long term. Indeed, AFM1 is known for its carcinogenic properties, and its chronic consumption has been associated with the development of human hepatocellular carcinoma, so HCC. And the HCC potency of aflatoxin is enhanced in individuals with hepatitis B infection. Consequently, most countries have set maximum limits of AFM1 in food. In Lebanon, the maximum limit in milk for AFM1 is based on the European limit, which is 0.05 microgram per liter of milk. In addition to the establishment of maximum limits, continuous monitoring of contamination levels is also important to control these food safety hazards. Indeed, hazard and risk are two different things, and just because a hazard is present in foodstuff does not necessarily mean that it represents an equal risk for the different subgroups of the general population, like, for example, the elderly, the teenagers, or the children. For that, we need to conduct a risk assessment first. And to perform a risk assessment, we first need to determine the exposure dose through food, which take into account AFM1 occurrence and concentration in foodstuff, and the consumption of the foodstuff in question, in our case it's milk, and the body weight of consumers. That dose is expressed in nanogram per kilogram body weight per person per day. For all these parameters, we need data. And the data we have at our disposal will have a huge impact, in fact, on the type of risk assessment that we can carry out. But I'll explain in more detail on the next slide. Once the exposure dose has been determined, it is possible to calculate the risk incurred by a population through the consumption of a particular food, for, in our case, again, it's milk, with regard to a specific health effect. For example, you can calculate the impact of AFM1 on the number of specific cancers. In our case, we calculated the number of HCC cases per year per 100,000 population that was caused by AFM1 exposure through milk. It is also possible to use other approaches to quantitatively estimate the risk, like the hazard index or the margin of exposure approaches. In our case, we used the margin of exposure to estimate the risk through the consumption of milk. So obviously, a general population is composed by different subgroups that may face different levels of risk due to intrinsic variables such as consumption or body weight. I'm sure you are well aware that a child doesn't have the same consumption pattern than an adult, nor the same body weight. However, if you use mean values for the different parameters in your risk assessment, you will only have yes or no answer. For example, FM1 concentration levels does not result in an adverse health effect for the general population. But if you want to estimate the risk for different subgroups, you will need non-aggregated data, meaning distribution for the different parameters, which means access to individual data points. This may enable us to identify at-risk population subgroups or consumption habits that would have been masked otherwise. A risk assessment does not necessarily need the generation of new data every time. Data that has already been published for other purposes can also be used. For example, you can use the same milk consumption in Lebanon to assess the risk for different hazards, for example, for pesticides, antibiotics, POPs, heavy metals, etc. Sometimes it is in fact just a matter of data collection. A lot of data that we used was previously published and even used in risk assessment, but exploited in aggregated form, given limited insight. In our case, we used published data, but used them in a non-aggregated form to allow us to identify the risk for subgroups. But of course, we also developed and published new data that had been missing, such as a data set of teenage milk consumption or data set for adult and adolescent body weight. So in order to carry out a robust risk assessment, it is important to identify significant differences in the various parameters that will have an impact on the exposure, on the exposition, within population subgroups. For example, do we have different milk intake per age or per gender subgroup? Do we have differences in the body weight per age or per subgroup? Another one could have been, do we have different FM1 concentration in milk per region? But in our case, in the case of Lebanon, this wasn't relevant since we are dealing with national distribution. But potentially, it could have been useful to identify problematic production sites. The point is, if you are able to identify differences that could lead to different exposition, then it would be ideal to perform different risk assessments. And now I will hand over to Sylvia for the presentation of the methodology and the results.
Hi, everyone, and thanks, Jeremy, for the introduction. So in this part of the presentation, we will go over the risk assessment model in more detail. As Jeremy mentioned, an important preliminary step when we're working with non-aggregated data, so point values, not just a mean, is to conduct some basic statistical analysis to allow us to identify significant differences between subgroups of each variable. In our case, we're lucky to have non-aggregated data for each of the variables that determine exposure. So as you see in the equation here, these variables are AFM1 concentration in milk intake per person per day, and body weight, so the individual's body weight. In our case, for concentration of AFM1 in Lebanese milk, we could have looked, for example, at the data per region of production. Perhaps some regions in Lebanon had significantly higher levels of AFM1 for some reason. However, we considered it was not relevant to our risk assessment because regardless of the area of production, the milk would be distributed anyways at the national level, and our assessment is a national level assessment, so for the entire Lebanese population, not only for a specific region. So we chose not to do this, but this is an example of something that could have been done maybe for another purpose of a risk assessment. For milk intake, however, again, we had access to non-aggregated data. In this case, it means we had data stratified per age group. So we had adults and adolescents. and per gender or sex, so male and female. And after doing our statistical analysis, we found that among adults, males and females consume equivalent amounts, so not exactly the same, but statistically equivalent. But this was not the case for adolescents. On the other hand, when we looked at body weight, the weight of adolescent males and females was equivalent, but it was not the case for adults. So what do we do with this information? This means that we will fit each of these different sets of data to probability distributions, which are a probability distribution is a mathematical function that describes the probability of sampling a given value from the entire data set. And when we set up the exposure calculations in our risk assessment model, these distributions are the ones that are going to be used as input. So you see the equation for exposure for AFM1 concentration, milk intake, and body weight. We're not going to use a mean value. We're going to use randomly sampled values from the distributions I just mentioned. But we will go over that in a minute. Okay, so maybe with these figures, I can show more clearly the differences I just mentioned. The figure on the left in blue shows adolescents' milk intake. The means are shown with an asterisk, and they're relatively close, as you see. However, having access again to non-aggregated data allows us to see that the distribution, so the entire length of the box plot, all the data points, are in fact different. So the groups are different, and that male adolescents' milk intake is overall larger than that of females. On the right, in red, we have body weights. Adolescents are shown in one single box plot because we did not find significant differences between genders. But for adults, again, we can see that males with a mean of more than 80 kilos have a significantly higher weight than females, which are around 70 kilos on average, let's say. So again, the means are shown with an asterisk. And we need to incorporate these differences in our risk assessment if we want to be able to produce estimates as accurate as possible for each of the population subgroups. So those were the variables that determine exposure. For risk, we estimated the additional numbers of HCC cases per 100,000 population that could be attributed to consumption of milk contaminated with AFM1 as the product of exposure and AFM1 cancer potency. So this is the equation that allows us to calculate additional HCC cases. HCC, Jeremy explained before, is hepatocellular carcinoma. So exposure is actually an output of our risk assessment. We calculated based on the variables we just explained. Cancer potency, on the other hand, is an input. We need to provide this number. So where do we get this from? Previous Lebanese risk assessments used the value of 0.0083. This is based on JECFA's estimates for non-European countries. So this is not specific to Lebanon, it's just non-European countries. And it assumes 25% prevalence of hepatitis B virus among the population and a standard body weight of 60 kilos. We can already tell that body weight of 60 kilos is not going to work in our case, based on the data I just showed you in the figure just before. And also, the prevalence of hepatitis B virus in Lebanon is much lower than 25%. It's actually only 1.74%, according to a study from 2016. So we can definitely fine-tune or adjust this value to be more representative of the Lebanese case. So what we did was, instead of using this JECFA universal kind of non-European value, we used a more recent AFM1 cancer potency estimate than the one from JECFA that was from 1999. So we used one from FAO WHO 2017, which is based on AFB1's potency. And we adjusted it considering the prevalence of hepatitis B virus in Lebanon. And this results in a smaller value than the one used in previous risk assessments. You see, we used a value of 0.00579. So also, if we had had access to prevalence of hepatitis B virus in different population groups in Lebanon, we could also have actually gotten more specific and produced cancer potency for specific subgroups of the population. But we did not find this type of data. And in any case, Lebanon has a very strong vaccination policy, so significantly higher prevalence values of hepatitis B virus were really not expected. Okay, so now that we went over the input variables, right? We went over exposure and we went over risk. We would be ready to set up our model. But first, let me explain just quickly how a probabilistic assessment works. So as we keep saying, our variables are described as distributions, not as point values. And you can visualize this perhaps as the figure here on the right, the blue curve. This is a distribution, and as you see, it's not a normal curve, not a bell-shaped curve. It's skewed. This means that most values in this case are towards the left, meaning that the majority are small quantities. But we also have some data points representing larger values on the right. This is typical of consumption data, food consumption, concentration of the hazard in the food. So most of the variables that we will use in risk assessment will be skewed. We're not necessarily going to be nice bell-shaped normal distributions. In our model, for each of the population groups considered, so we saw adolescent males, females, adults, males, females, we will simulate 10,000 individuals, each with a specific body weight, ingesting a given amount of milk that is contaminated with AFM1 at a certain level. So this will be the case for each of the 10,000 individuals in the simulation. The Monte Carlo simulation is programmed such that for each individual, a random value of each of these variables, so milk intake, milk contamination, and body weight, is selected from the corresponding distribution and combined to calculate a given exposure. You can imagine this process may be easily as that for each variable, we could represent it as a bag full of balls, and each ball will have a given value. So then for each iteration, a ball will be randomly sampled from each bag. So this bag will be the distribution, and these values will be then used to calculate exposure. And then we repeat this process for each of the 10,000 individuals. So we have 10,000 exposure doses at the end, and we will use these doses to calculate 10,000 HCC risk values. And then we repeat this, actually it's not over, we repeat this 1,000 times. So we have 1,000 groups, each including 10,000 individuals. So as you can imagine, at the end, we have a lot of data, and with all this data, we're not just going to report a mean value, but we're going to also create a distribution, so a range of outcomes, depending on the characteristics of each individual's exposure. Or you can also see this again as filling up a bag with each of the exposure values you produce, and then we use them to create a distribution. So I hope that made it a bit clearer. In this table, you can see in very simplified terms how the model is set up. So we have our variables on the left, AFM1 prevalence in milk, AFM1 concentration in milk, etc. And our outputs with checks, exposure and HCC risk. And on top, we have our population subgroups, so adults, females and males, and adolescents, females and males. You can see that there is no differentiation among groups for, here in the top rows, for AFM1 prevalence in milk and AFM1 concentration in milk. The same function is used for all cases because, as we said before, milk is nationally distributed and can reach any individual in the simulation. For daily milk intake, we have a distribution for adults, and 2 for adolescents, depending on the gender. And for body weight, we have 2 for adults and 1 for adolescents. This is based on the significant differences we found in our preliminary analysis. And by the way, I only added the names of the distributions here, so log normal, Weibull, gamma, etc. But even if you see like 1, 2, 3 log normals here for body weight, for example, each of them has a different set of parameters that describe them, so they're not the same equation. They follow the same distribution, but they're not the same exactly. Okay, so then we calculate exposure for each group, so each of the subpopulation groups, using the common AFM1 cancer potency that we just described before, and then we reach our HCC estimates. That was a quick overview of the model. Let's go straight to the results now. We won't go over the computations in this presentation, but you can find all the details in the article, okay, which is open access. So we obtained exposure distributions, exposure dose distributions for each population subgroup. So these are the subgroups. We have here the description of the distributions in terms of the minimum values, the 25th percentile, the median, which is the 50th percentile, the mean, 75th percentile, and the maximum value of the distribution. So as you can see, the range is very wide, right? We go from 10 to the minus 5 to single digits. And notice also that the mean values are actually larger than the median, which is typical of skewed distribution, like the one you see here, for example. In this case, our distribution, well, is way more skewed than the one in the figure, because 50% of values are very small. They're lower than 10 to the minus 4. So you can imagine why providing only mean value for this type of data is not very informative, right? But let's say we look only at the means. We see that adult males face the lowest exposure doses and adolescent males the highest. So all that is great. We have numbers for our exposure doses, but the important question now is, can these exposure levels result in adverse health effects? Or, do we need to be concerned with these numbers? What do they mean? So to answer the question, we will use 2 approaches. One is estimating the HCC risk that we mentioned before, which is the number of additional HCC cases per year per 100,000 population. As expected, the data is also very skewed, but less so than exposure. And I said expected because, as you remember, we calculate HCC risk using exposure in the equation. So there's an alignment, right? It's inevitable. So these results indicate that adult males would have the smallest number of additional HCC cases, and adolescent males the highest. And this was expected, again, as risk is aligned with exposure results. But notice that adult males can actually reach the highest max values. And this is probably linked to extreme consumption events, like somebody that consumes a lot of milk in one day for some reason. So this is still captured in the risk assessment when you use non-aggregated data. So with these results, we can now characterize and also rank the risk, right? Who has the highest risk? But again, should we be concerned with these values from a public health perspective? So to answer this question, we'll use another approach to characterize risk, which is the MOE, the margin of exposure, which is the ratio between a low-risk benchmark dose of the hazard from toxicological studies and the estimated exposures. So I see it's not on the slide, but it's just a simple division. Is this benchmark dose from toxicological studies divided by the estimated exposure? And if this ratio is lower than 10,000, it is generally considered of concern. So our simulations show that adults face limited risk since less than 25% of the calculated or the estimated MOE values obtained were below this threshold. Specifically, as you can see on the small table on the right here, the likelihood of being exposed to a dose of concern is between 17 and 20% among this population group. So not that high, really. For adolescents, here, the distribution of MOE values had a mean, so already a mean. We see the mean is below 10,000 for both males and female. So this could be already concerning. However, we know this distribution is very skewed, and the mean may not be the most useful statistics. So if we look at the likelihood of being exposed here, the likelihood of being exposed to a dose of concern for adolescents, this value was between 29 and 30%. So we're going to say 30%. These levels are starting to be more concerning, especially considering that adolescents, from a public health perspective, being a younger population, are a more vulnerable population than adults. So although we don't have a definitive threshold for what's risky or not, this example, this work showcases how this type of study can inform risk management decision way more than a simple deterministic assessment where you would get just a mean value for everything, for the entire population, and that's it. And this, we also try to show how we can fine-tune our model to identify specific populations at risk, but also specific populations that may not be at risk. So in this specific case, although we know AFM1 is indeed a contaminant strongly associated with milk, we actually demonstrated that despite some non-compliances reported in the literature, Lebanese adults are still well protected by the control measures in place. So we may need to focus more on younger populations when we talk about risk from exposure to AFM1 through milk. We should not generalize it. Everybody is at risk. This is a terrible hazard. I mean, it's a terrible hazard, yes, but not everybody faces the same level of risk, and especially adults seem to be facing limited risk. So it's interesting that we are able to focus and identify, fine-tune which are specific groups that may be at risk. So I'll stop now and let's go back to Jeremy to conclude the presentation. Thank you.
So thank you, Sylvia, for the presentation of the results. So before returning to our results, there are some general remarks to be made, not directly related to our results. The first one is to update your risk assessment as soon as new data become available. And the second point is, I really hope that we have convinced you of the value of non-aggregated data for conducting your risk assessments. It will enable you to obtain much more information and to put measures in place in a much more refined ways. Now, if we look back at our results, they showed that adults are at low risk in Lebanon, and adolescents seem to be slightly more at risk. Considering that AFM1 exposure from other sources, like, for example, dairy products, is expected to increase this risk, reduction of AFM1 contamination in milk in Lebanon should be a priority for risk management efforts, especially aiming at protecting younger populations. This study also highlights the fact that there is a need for similar studies in younger populations, children or babies, which would require the collection of representative individual level milk intake data. So that's the end of our presentation. If you have any question, I invite you to ask Samuel, who must be probably in the room right now. Thank you very much and have a very nice day.
Well, our thanks and my thanks in particular go to the team in Quebec for preparing this presentation, in fact, last night and sending it. I hope actually that through this illustration and really repeating some of the key messages that were shared by the team there is that, first of all, I think we are reiterating the importance of data. Data, 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 that's really what is really important. We saw here that if we made the decisions on the basis of means, we would have made a wrong risk characterization. We would have identified risks where risks did not exist and potentially underestimated other risks. So deaggregated data is absolutely key. And to have deaggregated data, we need to have the information from you, individual data points. The other, I think, key message as well is that even for the hazard characterization that is available from international reputable sources, we cannot take the information for face value. We have to have discernment to examine this hazard characterization information. In that particular case, JECFA had an assessment that dated back to the 1990s. They went back and looked at the data and put it in the context of the Lebanese situation in order to identify the potential hazard characterization values that they need to rely upon for the risks that they considered, which is essentially an increased risks of hepatocellular carcinoma. So again, they used things in the literature, but they used the appropriate source of information from the literature. So these are some of the key messages, again, that we wanted to share with you. And of course, hopefully this will be feasible in various parts of the Arab region with the availability and particularly not only the availability at the local level, but the availability of shared data points. So essentially the possibility to access that data. I take it that there are no questions because I'm not. Oh, there is. So I tried to have a long conclusion so as to avoid questions, but I didn't succeed. Please. Oh, we're going to take a microphone, Engineer Shahada. And I would like to mention actually also to our colleagues that this event is run in a hybrid mode. So people are hopefully following online, but also on YouTube. And to mention that Arabic interpretation is also available. To access it, however, in the room, you need to link to the Zoom connection. Colleagues who are online are accessing the Arabic interpretation.
Okay, thank you, Dr. Sam. Actually, it's a very important issue. This presentation, go directly regarding to the aflatoxin M1. If we compare the EU and the Codex, we found the raw milk is the item that's evaluated. The Codex is 0.5, while European is 0.05. So it's 10 times. This is for liquid milk. I think the presentation talk about liquid milk, not dehydrated milk, because if we follow the dehydrated factor, it should be 10 times from liquid. That's true?
Yes. Yeah. I mean, when we try to get the ml, yes, we try to estimate the correspondence between the different matrices.
So European, for example, put
however, Engineer Shahada, what I would like to say, that is always an approximation. So in the sense that when we have an ml for a commodity that is different from the commodity that we are targeting, we try to make the correspondence when the correspondence is not available, when there is no ml that is available. However, that's an approximation because we're not sure that that's the same type of exposure, meaning what is the level of consumption and use of dehydrated milk versus the liquid milk. So it is an approximation. And of course, when we make that, we have then after that to characterize that approximation, meaning how likely it is or how not likely it is, in order to see the result and look at it really with discernment. So that's why that tenfold factor is on the basis of approximation. When I say approximation, meaning that we are making the hypothesis that people consume in the same manner, households buy in the same manner liquid milk and dehydrated milk. That's an assumption. Is it true or not? That's essentially what then you look at the answer on the basis of that. Sorry.
And for dried milk, you should evaluate after reconstitution. So it's the equation will be true. Number two, we have or we can use such model in other food product. And for example, we have the average of consumption, meat consumption worldwide is around 100 kilo. In Egypt, for example, it's around 50% from such number. So the contaminants in meat should be evaluated as per consumption, as mentioned here, because the exposure is different between, we'll talk in Egypt about this. Thank you.
Yeah, absolutely. I think, I mean, when we do exposure assessments and risk assessments, we do things on the basis of what is consumed. Risk management decisions, however, are generally based on commodities as sold. So that's because, of course, we're trying to make the risk management decision easy to enforce. So we make it on the commodity that is sold.
Yeah, I will have a quick one just regarding, the clinical studies or the process that they normally use in such a research. How they gather, a group of human and they expose it to certain level of M1 aflatoxin and they can measure, the risk related to this one. Or this research, it might be done, in vitro or something like that.
So in this particular case, it's actually based on epidemiological information. So not necessarily, this is not really on the basis of animal studies or situations like that. This is really on the basis of epidemiological information and then determining a risk factor for the potential increase of cancer. So in this particular case, it's an increase of cancer risk. So basically, you have an exposure to 1 nanogram per kilogram body weight per day. So you set the level of exposure, and you have an increase of 1 cancer per 100,000 people. So that's essentially what you're trying to set. So you set a level of protection with the cancer risk that is, I don't want to see an increase of more than 1 cancer risk per 100,000. It's the slope of a cancer risk determination. And that's generally obtained from epidemiological information.
Thank you.
Thank you very much for the presentation. My question is very basic especially for the developing countries. Because particularly for mycotoxin and aflatoxin, different countries have different regulatory, limits in foods. For example, North America, like Canada and America, had got 20 ppb for aflatoxin B1. And for N1, like the colleague said, 0.5, and for EU is 0.05. Similarly, in European countries, the level is very low. Is it based on risk assessment or economics?
So generally, risk management decisions are based absolutely on risk assessments. But the risk assessments can be different from one country to the other for different reasons, for various reasons, various parameters that can actually explain the differences in risk assessments. One, of course, is related to the exposure. And the exposure comes from the 2 factors: differences in the occurrence level, differences in the consumption patterns. The difference in risk assessments also can come for or as a result of differences of the level of protection that is accepted by a particular jurisdiction. And that's the prerogative of each jurisdiction to determine the level of protection. So one country can decide that, for example, I gave you the example, is for 1 nanogram per kilogram body weight per day, I do not want to see more than 1 cancer increase per 100,000. You may have another country that say, you know what, that level, I want it to be 1 in 1 million. I want to protect more. And that's a decision of the country. to make. You see, this level, this is what we call the acceptable level of protection, the ALOP. And this acceptable level of protection is part of what we call the risk assessment policy. Each country, based on its risk management decisions, can set that acceptable level of protection. And that's why you may have differences in that regard. It doesn't mean that the risk assessment is wrong. It's just that the parameters that are used or the data that are used may be different, which actually reemphasizes the idea that when we have a risk assessment that we want to use or to rely upon, we need to look at it with discernment to see its applicability to the jurisdiction that we are trying to make that risk assessment for. I think, yeah, for the interest of time, we will stop here, and I'm sure that we will continue to have some discussions about this later on. Now we're going to actually practically stay in the same theme, where how do we treat data and information coming from various jurisdictions? And we're going to be examining the case of titanium dioxide. Titanium dioxide is a food additive that was subject to risk assessments from different jurisdictions. The European Food Safety Authority came up with an assessment, and then it was followed suit by a number of other jurisdictions, and particularly the Joint Expert Committee on Food Additives of the FAO-WHO, which is the advisory body of Codex. And to discuss this particular case, applying evidence-based approaches to manage titanium dioxide, I would like to invite our colleague, Dr. Riem Gissouma, one of our colleagues in the G-Force team, but also a member of the National Food Risk Assessment Agency in Tunisia.
Good morning, everyone. Thank you for joining this session. Today, I present myself. We am Gissouma, expert in G-Force. I'm from Tunisia. Today, I will present the topic on applying evidence-based approach to manage titanium dioxide as a food additive. Okay. Okay, titanium dioxide, also known as E171, is a mineral used for its bright white color and opacity, widely used in some food product as confectionery and dairy product, baked goods to enhance visual appeal. Also found in non-food application such as cosmetic and pharmaceutical use. Recent studies and assessments have raised safety concern, especially regarding nanoparticles risk. Derives regulatory response worldwide reflecting weighing approach to manage titanium dioxide use. So the source of exposure to titanium dioxide in food coming from intentionally as a color additive and intentionally from leaching from plastic and other packaging materials or from agriculture input, fertilizer, pesticides, and also from environment contamination through irrigation water or contaminated soil. Historically, titanium dioxide has been used for decades, considered as safe when applied as a food additive. However, recent assessments by EFSA, European Food Safety Authority, on its nanoparticle components, which led to concern about possible genotoxicity effects and DNA damage. So the nanoparticle, smaller than 1 nanometer, There is concern that they can penetrate human tissue and cause possible genotoxicity risk and potential DNA damage, with possible accumulation of nanoparticles in human tissue. That presents a risk to vulnerable population that consume a high quantity of confectionery and dairy product children due to higher consumption of sweets. So the food grade application typically for titanium dioxide is between 200 nanometer and 300 nanometer. But with food grade, in some case, we found nanoparticle with size smaller than 100 nanometer. So, referring to some studies from various regions, including US study, European, Chinese have examined dairy exposure to titanium dioxide nanoparticle. This study shows highlights that the particle size of E1T71 is typically range from 200 to 300 nanometer. However, the key found from this study, generally we found between 10 to 36% of these additive particles are smaller than 100 particles. Other study also for unintentional exposure to titanium dioxide illustrate how these pathways contribute to presence of titanium dioxide in food. So the titanium dioxide contamination level in food coming from unintentionally can range from 0.01 to 1 milligram per kilogram due to environment contamination or leaching from packagings. Here, there are a comparison between EFSA and the JECFA approach. There is a clear divergence in how different regulatory bodies manage titanium dioxide. JECFA and EFSA have taken notably different approach. EFSA, however, internationally, has Canada, US FDA, Australian and New Zealand Food Safety Agency, Japan Food Safety Commission disagree with EFSA assessment in line with JECFA. For EFSA also is placed move emphasis on nanoparticle risk with the study where titanium dioxide nanoparticle proportion is not representative of food grade titanium dioxide. For RFDA, no relevant for EFSA given the concern expressed. All other agencies concur with JECFA. For the effects of titanium dioxide, JECFA found no cancerogenic or reprotoxic risk, while EFSA emphasizing possible genotoxicity concern as a result of the considerate of nanoparticle proportion, not representative of food grade application. Other agencies concur with JECFA. For dairy exposure, JECFA selected a high estimate of 10 milligram per kilogram body weight per day for dairy exposure to A171, focusing on low absorption and lack of identifiable hazards. So here we found that EFSA declared E171 as unsafe as a food additive due to concerns over potential DNA damage and possible genotoxicity leading to being in Europe. While JECFA maintained approval of E1T71 and calling for more research with no immediate ban implemented. So here we recommend for countries relying on EFSA assessments with titanium dioxide banned as a food additive to identify safe and equal well-studied alternative to this food additive as whitening agent, ensuring they offer similar functionality with associated health risks. And second, they have to develop regulatory cutoff level for compliance verification and to differentiate between intentional for food additive use and unintentional environment presence. So the food additive use is banned versus environment presence possible at various levels. Here we present the approach, development of practical approach by Food Safety and Quality Center in Sultanate Oman. With presenting the approach here, we can find the description, statistical description and distribution of titanium dioxide in food. The value, the level, the cutoff level defined is 25 ppm. So the level when is below 25 ppm, we rule out intentional addition of titanium dioxide as a food additive. But when the level exceeding 25 ppm, would initiate an investigation to define if titanium dioxide was used and used intentionally. For countries that concur with the JICFA and Codex direction, we recommend to continue vigilance on safety data, research bioavailability and accumulation, review the specification to ensure low nano level, and conduct comprehensive toxicological studies, improvement in detection technology, mainly for nanoparticles, and guidelines for good manufacturing practice, mainly for food grade application, and enforcement to tire control on additive food grade specs. Finally, the result of studies showing concern were obtained with material that does not represent food grade applications. Thank you for your attention. I give you the floor for any question. Thank you.
Thanks very much, Riyam, actually, for summarizing a very complex issue in very few slides, because it's a matter that took quite a bit of time. I would like to add actually a couple of points on Riyam's great presentation here. Titanium dioxide is a whitening agent. To obtain white, you need between 400 and 700 nanometers. That's essentially, to have white, you need between 400 and 700 nanometers. When you have nanoparticles, it is 100 nanometers and less. 100 nanometer and less brings us to UV, meaning that we are transparent. Which means that if you're using a material that has a high proportion of nanoparticles, that's not your whitening agent. It's basically not food grade. It means that it is a product that is not fulfilling the food grade objective, because why would you use this as a whitening agent when it's not giving you the white? So the issue in this case, it's not that there is a dispute about the studies that were used upon. In this case, the review here is the relevance of the results of the study for the application that we have. If you have a product that is not fulfilling the food grade specifications, you have a problem of non-compliance with the specification. That's not actually that there is a problem with the food additive. It's just actually when you say that I have a food additive that is prepared outside of the specification of the food additive, it's a polluted food additive, and I'm saying I have a problem with the food additive. No, your food additive is not fulfilling the specifications. at least this is the explanation that, or one of the explanations. I'm trying actually to put it in simpler terms. I'm not a toxicologist myself, and to be honest, I relied on the support of Dr. Mark Feely, who in fact is one of the experts that coordinated the JECFA review, the latest JECFA review. And he essentially, and by the way, I invite you to listen to the webinar, to watch the webinar that GeForce organized a few weeks back on this very issue, and that I think we captured the essence of, because it was more than an hour webinar, where essentially we went through the entire process of the EFSA assessment and the JECFA assessment, and trying really to make the discernment between those two elements. So again, these are all illustrations about how a risk assessment is to be used, and even actually in this case, how different jurisdictions using different risk assessment approaches, and how essentially the fact that one jurisdiction comes out with a conclusion, the other jurisdictions have the possibility of reviewing those conclusions. And in fact, when EFSA issued its report in, I believe, 2022, that's I think what it was, in 2022, basically Health Canada did an assessment, the US FDA did an assessment, the Japanese Food Safety Commission did an assessment, the Food Standards Australia and New Zealand did an assessment, and they all actually had differences with the conclusion of the European Food Safety Authority. But what is the culmination? Because at the end of the day, we are all behind Codex, and we are also behind the advisory bodies of Codex. JECFA was tasked by the Codex Committee on Food Additives to conduct an assessment. The JECFA conclusion came out most recently. In fact, it more concurred, and I think VM summarized it very nicely there. It concurred with the other jurisdictions' conclusions. This does not mean, again, that we should not have any vigilance in the situation. This means that, like with any situation, this is a scientific-based process, and we have to follow the science and the evolution of the science, including the evolution of the interpretation of the science, and basically take decisions on the basis of that. So hopefully that puts in context the situation.
We are now, I do believe that we are in two minds. Which one we have to follow? Because since 2022, EFSA issued, the ban of using the E171, the titanium dioxide. So all the Arabic countries, one after another, they start to ban it as well. Keeping in mind that they are following Codex as well. Even, in Egypt, we are following Codex as well, but still till now, we did not ban it based on the final JECFA assessment that they have. So from your angle, which direction that you would like us to take?
So I would like to say first that every country is sovereign, and every country is able and has the responsibility and even the prerogative to make a decision that fits their requirements. After that, if they get questioned about that, it's up to them to provide the evidence or the response in that regard. Now, my response to your question more precisely is, if we want to follow what is the latest decision by Codex, the Codex Committee on Food Additives, at its 54th session, which met this year, so essentially we're talking here, I believe it was March or April, anyway, it's this year, it was March, right? March 2024, reviewed the latest assessment by JECFA and decided to keep the authorization of titanium dioxide as an approved food additive, with the indication that JECFA indicated that we have no ADI, the ADI is not specified. What does this mean? Is that there is no concern, which means that the level of use is at GMP, at good manufacturing practice. So Codex, the CCFA, came up with this. I believe, in fact, this also conclusion will be sent to the Commission. I don't think that the Commission will change anything, because this is really maintaining a decision on the basis of what JECFA did. Again, also, when you look at the JECFA reports, like any risk assessment report, always the conclusion is this is what we have with the latest evidence to date. We keep vigilant. We keep always up to date with any type of changes in the scientific evidence. And if there is a change, a scientific assessment will be updated. But at this point in time, from an international perspective, both JECFA and every other jurisdictions that we mentioned, from Canada to the US to Australia and New Zealand, are not in concurrence with the concern that was expressed by the European risk assessment. I think we've exhausted this subject. Now we're going to move to another topic. So you see, this session is really about illustrations of various risk assessments. Another area of interest are now risk assessments about regulated products. And we started the case with the titanium dioxide because that's a regulated product, and this warrants a safety assessment. Now we're going to take a slighter shift from a risk assessment towards looking at the evidence to develop risk management approaches. And we are fortunate to have Dr. Rasha Galal from the National Food Safety Authority of Egypt to share with us the Egyptian approach to develop a risk-based management approach for energy drinks in food. Dr. Rasha.
Good morning. I'd like first to present myself. I'm Rasha Galal. I lead the team for regulating functional food at the Egyptian National Food Safety Authority. I'd like to thank Dubai municipality for giving us this opportunity, and of course, Professor Samuel for helping us develop this risk assessment approach for the regulation of energy drinks. First of all, a monograph for caffeinated energy drinks was developed by the National Food Safety Authority of Egypt as the basis for approval of formulation of energy drinks destined to be marketed in the Egyptian market. And this is a growing trend in Egypt currently. Every day we have more and more energy drinks in the market. Most of them are regulated, but every now and then we tend to find products in the market which are not yet regulated. This monograph includes set formulation and labeling provisions, which will serve as the basis for rapid review and approval of such products. This presentation will offer the evidence-based justification of the decisions reached by NEFSA regulators supporting the energy drink monograph. The monograph is based on the review and adaptation of previous assessments which were done by reputable international bodies such as EFSA and Health Canada. And we mainly depended on the data presented by these authorities. So what is an energy drink? It's a non-alcoholic beverage, carbonated or not, that contains a higher level of caffeine, which is the main component of energy drinks. It contains higher caffeine than other soft drinks, with the purpose to induce the effect of alertness associated with this type of ingredient. These products can be flavored or not and may include other food ingredients commonly present in food in the Egyptian market. The first risk we handled was the portion size. When thinking of what portion size to allow, we reached the decision of 250 milliliter portions. So what was the risk? Most of the safety assessments associated with caffeinated energy drinks have uncovered that the risks associated with the possible overexposure to active ingredients in these products, such as caffeine, are mainly attributed to the consumption behaviors rather than the actual composition of these products. So it's the overuse sometimes of these products that make the risk. How did we mitigate this risk? That we decided that a portion control of such types of products would be a reasonable way to mitigate the risk. So this measure was recognized as a suitable approach of risk mitigation, contributing to possible lower consumption of the beverage. And as mentioned, it was decided that a standard 250 milli portion would be suitable for these types of products, while larger products may be assessed separately. The second risk was consumption. So it was decided that 2 servings per day was a reasonable approach. As mentioned previously, that the risk of these products was mainly the overexposure to the active ingredients rather than the use of these products. The mitigation was most risk assessments carried out by food regulators internationally concluded that the consumption of 2 servings of a typical energy drink product modeled on a 250 milliliter was considered safe for the general population. The third risk came from the composition, and the main composition of energy drinks, as mentioned before, was caffeine. So to assess the risk coming from caffeine, we made a hazard assessment and an exposure assessment. The hazard assessment of caffeine was mainly that it acts as a stimulant, may produce mild side effects like anxiety, headaches. Some people are sensitive to caffeine more than others. Stronger effects when higher amounts are absorbed quickly. However, most of the effects are transient and ceasing when stopped taking any products containing caffeine. And it's not considered to be clinically addictive. And so it's possible to determine a safe non-adverse intake level for caffeine. As for the exposure assessment, most of EFSA reports indicated that over 1/3 of caffeine intake for the European population is associated with consumption of coffee and coffee products, while this percentage is higher in the USA. Well, it was reported at about 64% of the total caffeine intake. So when determining the suitable amount of caffeine in energy drinks, we used a conservative approach. So safe levels for caffeine are around 400 milligrams per day for adults, which is equivalent to 5.7 milligram per kilogram body weight per day for a 70 kilogram healthy individual. As for children, it is safe for around 2.5 milligrams per kilogram body weight per day. And this level is around 200 milligram per day for EFSA and 300 according to Health Canada for pregnant women. The safe intake of caffeine for children and adolescents has been set at levels varying between 2.5 milligram per kilogram body weight per day and up to 3 milligram per kilogram body weight per day. Adopting such low safety levels initially derived for children to adolescents is considered a very conservative approach and therefore quite protective for the general population. So it was decided that a 400 milligram per liter in energy drinks with a minimum of 200 milligram per liter to differentiate them as functional food or special foods. For volumes higher than 250 milliliter, a maximum caffeine amount of 180 milligram per container is also set, which is corresponding to a small cup of coffee. And this was considered as a conservative approach of allowing caffeine and could be recommended for consumptions by those 16 years or older. Such level would ensure that a consumer would not exceed the ingestion of more than 100 milligram of caffeine in one consumption setting, and this would correspond to a level varying between 2.5 milligram per kilogram body weight up to 3 milligram per kilogram body weight for somebody weighing between 70 and 60 kilogram body weight. Other typical components of energy drinks, which are usually, when we surveyed the energy drinks in the Egyptian market, we found that most of them contain taurine. So we decided that 1,000 milligram per 250 milliliter serving, based on guidance that occasional consumption of up to 2,000 milligrams is considered safe. So having a maximum of 1,000 milligrams would be considered as a safe amount. As for glucuronolactone, it will not exceed 600 milligrams per 250 milliliter serving. For inositol, it will not exceed 200 milligrams of inositol for a volume of 250 milliliters. Coming to the next major components of energy drinks, which are vitamins and minerals, specifically vitamin B. We used a methodology which was adopted by Health Canada, and we used it to apply to determine the maximum amount of vitamins and minerals to be used in these energy drinks. So the formula we used was determined as follows. The maximum amount of any vitamin or mineral would be formulated as follows: the safe daily amount minus the daily food and supplement intake over 5. The safe daily amount, which is the tolerable upper intake level of any vitamin or mineral, which is the maximum safe amount to be ingested daily. The daily food and supplement intake would represent the RDA, or the daily allowed intake of any vitamin or mineral, over 5. And when we chose 5, we thought that would be reasonable for the Egyptian population. That represents 5 times that the population can take any of these vitamins from other sources other than this energy drink. So we used this methodology for assessing the maximum requirement for vitamins and minerals, and we reached these figures for the different vitamins and minerals. Other restrictions in caffeinated energy drinks formulations: they must be formulated as a functional food without any therapeutic or hormonal ingredients. They also must be non-alcoholic with a maximum amount of alcohol not exceeding 1.1% volume per volume. Certain ingredients are prohibited in any energy drink. These include vitamin A in the retinol form due to its teratogenic effect, folic acid and its salts, nicotine and other herbs which might have any therapeutic effect. And NEFSA may consider evaluating and approving additional ingredients separately as provided by the owner of the energy drink. And to ensure that all caffeinated energy drinks are targeted at adults and adolescents 16 plus, any formulations which are known to appeal to children are restricted, so no milk or dairy ingredients are allowed in the caffeinated energy drinks. Fruit and vegetable juice should not exceed 25% of the composition, and the term juice cannot be used in conjunction with the name of the product. Other restrictions on the labeling. it must be mentioned clearly on the label that a consumption of more than 2 servings per day is prohibited, and the product is not recommended for children or anyone less than 16 years of age, and it's not allowed for pregnant women, lactating mothers, and individuals with known sensitivity to caffeine. And to further support the risk management measures enabling the safe consumption of these products as functional food, other labeling restrictions are to be observed, including the prohibition to include the word juice, as mentioned before, in the name of the product, including the use of the wording juice drinks or energy juice, the prohibition to have any health claim identifying the product as a hydrating or a source of electrolytes, and the prohibition to identify the product as water, part of the name of the product, and also prohibiting any claim that marks the product as a sports drink or an enhancer of physical or sports performance. Other risk management measures, the risk and mitigation strategies for caffeinated energy drinks, they include both regulatory measures and non-regulatory measures. For the regulatory measures, as we just presented, we restrict the formulation requirements and set labeling obligations. As for the other non-regulatory measures, there is a plan to have targeted consumer information and education campaigns, collaborate with health professionals, youth, and educational organizations, and provide information of energy drinks composition, conditions of use, and healthy consumption, and follow best practices in food risk management, integrate food standard development, regulatory measures, and non-regulatory interventions, align with the World Trade Organization recommendations, use risk analysis-based process to develop food standards, and regulatory measures justified by robust risk assessment and potential health risks. The main document was published on the Journal of the National Food Safety Authority last May, and it was open for comments until the end of August 2024. And we received a couple of comments on the document. The main comments received was an objection on size restriction. There was a request to broaden the list of ingredients which were discussed in the monograph and to include other vitamins and minerals such as biotin and some amino acids. And also there was an objection on not using milk or dairy products in energy drinks, and also an objection on prohibiting the term juice. Anyway, these comments will be discussed, will be reviewed, and if any adjustments need to be made, it will be done. As for the Egyptian market, this is a brief of the situation now in the Egyptian market of energy drinks. We have 36 energy drinks registered and licensed. 27 of them are important. 6 of them are produced in Egypt under license, and 3 local products. And recently we have registered 6 other local products which were already in the market without registration. And when we tracked the producer, we managed to convince them to come and register the product and adjust their formula to comply with this monograph. Thank you very much.
Very much, Dr. Rasha, for this great overview. And I think actually this is another illustration about how an assessment is being used. In this particular case, it wasn't a comprehensive risk assessment that was conducted locally, but rather for each element of the risk management consideration that was identified, basically an examination of the risk rationale for it. And of course, on the basis of risk assessments that were done by other jurisdictions in this particular case. So each risk management point that was mentioned, including the labeling risk management considerations, were all justified by a risk rationale in this case. So I don't know if you have any questions on this point. And in fact, on this particular presentation, or by the way, on the others, we're going to be opening, we have about 10-15 minutes that is really more of an open discussion. And the intent really behind these 10-15 minutes is really to discuss the horizons for risk assessment in the Arab region and in the GCC in particular. What are the needs? What are the interests? What are the impediments, the challenges? What are the enablers? Perhaps actually learning from each other's experiences in this regard. So happy to hear any input or any experience that one of you would like to share, or any question that you might have. Please, so we're going to have a microphone for you, so please.
Good morning. So we've seen the risk assessment and the maximum tolerable limit for the unintended use of TiO2. And this was specifically a requirement in Oman. So do we have a similar references in, I don't know, in JECFA or in the rest of GCC region to refer to in case of carryovers?
Okay. Professor Hossein, would you like to address that, or shall I address it? Okay. So actually, maybe a clarification. What was presented for the Oman example is not a risk assessment. That was an evidence-based support to making a cutoff for enforcing the ban. Because basically, Oman is a country of the GCC. The GCC followed suit with the EFSA assessment and decided to follow what the GSO decided, which is essentially that titanium dioxide is no longer allowed as a food additive. The question was, how do I implement that ban? And the problem is coming from where? Titanium is readily available in the environment and in food. And it is readily available in what form? In titanium dioxide. So when you try to do analysis of titanium dioxide, you're going to find it practically everywhere. So the question is, are you going to be applying whatever you find as the value for that ban? And the question that emerged is, the decision of the GSO was not about titanium or titanium dioxide as a result of unintentional presence or unavoidable presence. It is for the application food additive. You agree or you disagree with that decision, it's something else. It's how to enforce that decision. So the point that was raised by colleagues in Oman is, when I analyze products, I'm finding it everywhere. Where do I put my cutoff? Or how can I establish my cutoff in order to do that? So essentially, the approach that was followed was the following. First of all, there was quite a bit of analysis that was done in the Omani labs. So in the Food Safety and Quality Center, there was quite a bit of data generated. And there was a suspicion that for most of the data that was generated, it seemed to be more related to unintentional presence than the actual intentional presence. But it was a question mark. So the idea was, how can we establish that by looking at the literature? So essentially what the Omani colleagues did is that they surveyed hundreds of publications. They did practically close to a meta analysis of the information that is available. In fact, actually, the information is still being refined because the intent is to publish this approach as well, even though it was used from a practical standpoint. So the data was extracted from the publications, try to make the discernment between the data that is coming or that is clearly indicating that the levels are associated with a food additive use, the levels that are associated with natural or environmental presence, and some data was actually suspicious because it was not clearly indicated in the papers. So I'm going to spare you, the inclusion and the exclusion criteria. But essentially what happened is that they did a distribution then after of the data. And what they found was that most of the data for non-intentional use seemed to be around, I believe it was 2.7 parts per million, right? It was, I'm asking for the 70. Yeah, so basically most of the data, the 75th percentile, is definitely lower than 5 anyway. I'm sure that, it's very quite low. There were some outliers that were higher up there. And when you're looking after the correction of the outliers of the distributions, it was found that the maximum could be, or was found, in fact, at about 23 level. So they rounded that level and they considered that that level is a cutoff to investigate, which means that when I have that level, I consider that I have the right to question, is that coming from unintentional source or is it coming from an intentional addition, even if it was diluted? Like a carryover type situation here. And that, in fact, allowed to exclude, I would say, many, many samples from what was found. In fact, I think by practice, from what I recall of the data that was available, it was very clearly distinguishable between the unintentional presence, which is more in the 2-3 parts per million type or even lower, and the actual food additive use, which would be in the hundreds of ppm level. So that's why that cutoff of investigation was found to be a practical cutoff and also something that is enforceable. Enforceable for the intent of that decision, which is essentially to exclude the food additive use.
So much for the clarification. So based on this evidence-based approach, is it also recognized in the rest of the regions, like in Jordan or in the rest of GCC as well?
That, I think, is a question to be directed to the regulators from the region, not to myself. So, but I think this is an opportunity like another to disseminate the information, because I don't believe that information was widely disseminated at this point. I think one of the objectives is also to publish the approach that was made, so essentially to share it. And I think this is one of the objectives of a meeting such as this one, which is we exchanging the experiences, exchanging the approaches. As we said here, you may agree or disagree with the ban, but if you have to apply it from a practical standpoint, how do you apply it? Are we chasing ppm or sub ppm levels, or are we after higher levels? And in this case, what was found on an evidence-based approach is that we're talking about 20 and above.
Thank you so much.
Any other questions? It's basically we're right now discussing any topic of risk assessment, including interests in where we want to take things in the region.
No, I just wanted to use the opportunity to commend NEFSA on their work on developing this energy drink monograph, and we would hope that other regulators in the region would follow that example, so that because energy drinks are still a very a product that raises concerns, often based on outdated science, on rumors, and we would hope that other regulators in the region would follow NEFSA's example and develop a truly science-based standard. Thank you.
Thank you for that comment. Any other points? I don't know. I'm just going to maybe ask the regulators of in the region here, where are your current, I would say, priorities in risk management, whether be it, contaminants, pathogens, even regulated products? And do you have any challenges that you identify in managing those risks using, again, the risk analysis approach and risk assessment? Anyone would like to share their experience in this regard? Dr. Gouda, so just gonna have
Assalamu alaikum. Yes, I have some point related to risk assessment and risk management in the region. First of all, what is the priority? What is the priority for, as you said, for food contaminants? Which one we have to start? This is number one. Number two, a standardized approach for the region, because as you know, consumption in the region uh, most probably same. What is the priority for the contaminant? Is the pesticide, mycotoxin, veteran drugs, heavy metals, pathogen? Okay, if it's for example pesticide, which type of pesticide? Okay, which type of commodity we have to concentrate? There are some big issue related to regulatory limit, for example, in pesticide. For example, some of the commodity, there are no availability for maximum residual limit with a risk assessment study. For example, in grape leaves, no pesticide, no, some of pesticide, no specific maximum residual limit in Gulf, in Egypt, and even in the Europe, only precautionary limit like 0.01 for some pesticide. That's why, because most of this country, they didn't have consumption rate for the grape leaves, for example. What exactly approach we have to follow in the Arab region for risk assessment? From my perspective, I guess we have to have a standardized approach based on our consumption rate, based on our priorities, which commodity we have to concentrate for, even for pesticide or for any other contaminants, for any other, for example, pathogen. We need to standardize one approach which is can fit for all our consumption habit or dietary intake. Thank you.
Thank you very much, Dr. Gouda, for that input. I guess I think it will take probably a session just to try to answer every aspect of what you propose. But I think the point that I'm taking from your intervention is that it's very important that we establish priorities for management. And of course, the priorities can be established from different levers or different parameters that might be influencing that priority setting. It could be production conditions, it can be domestic considerations, it could be exposure situations, incidents that are faced by the jurisdiction. But I guess, the point that I would share with you is that we are, of course, all looking at Codex as a reference. And Codex actually, for the various horizontal committees that manage the risks. So for contaminants, it's the Codex Committee on Contaminants in Food. For pesticides, it's the CCPR. For vet drugs, etc. For pathogens, it's the Food Hygiene Committee. Each of these committees has actually set a prioritization approach based on the input of the entire membership. And that prioritization approach, even if it may not necessarily 100% relevant for every country, it has actually some relevance to most of the countries or to a certain level for each country. So using perhaps Codex as the reference in terms of priorities, because generally when Codex sets that as a priority, there are so many considerations that come as a result of that. There is a request for data. There is actually a request that countries contribute to the assessment that might be made by the advisory bodies, such as JECFA, JMPR, etc. So essentially, if those priorities are looked at by the community, the international community, including the regional communities, it would also prepare the region to be even more effective and more active in providing the data and in accompanying the process by which the international food standard setting process is being conducted. And therefore, also, it will ready the region to take stock of the output of the Codex decision and to see how it can apply to its own situations. Sorry, we're because of our colleagues in online, so just no, it's.
Yes. At this point, where we are in Codex, where we are as a region in Codex, how we share our data, this other question. If we are sharing our data, if we are have like recommendation. I was in one committee for I raised one big concern related to rice, not this year, last year, about the rice limit, and they asked me for data. Where is the data? I was representative for Qatar as I am consultant in food safety laboratory in Qatar at that time, and Emirates, Qatar, Saudi Arabian raised the same, but they told us you are very late because you are not related. Send your request officially during the channel. I am speaking about, cooperation between the countries in the region to let us in the codex, affect in the codex decision. This is my concern or my question.
No, thank you. Thank you for that. In fact, actually, I think, Dr. Gouda, you're making a great segue to the next session, which is going to be actually talking specifically about mechanisms of collaboration amongst food regulators in the region, including actually the determination of some of the priorities for that collaboration. So with your permission, we're going to use this as one of the starting points of the conversation we're going to have at the next session. So looking at the time, I think we've reached the end of this session. I would like to ask you to join me in thanking all those who contributed in preparing the session, whether be it in Canada or here, Dr. Riem Gisuma, Dr. Rasha Galal. Thank you very much for your contributions to this session, and thank you all for your active participation. And with this, we conclude this session on risk assessment. We'll take 5 minutes, if you don't mind. Technically, the next session is coming right away. We'll take 5 minutes for a health break, and then we will reconvene. Thank you.